Thank you very much, Peter, and it's a real pleasure and an honor to be here. Thanks for the invitation. So, today I'm going to talk about plant heat stress, heat waves and food security, and how those events are linked together. So what you see here are some very familiar events that are resulting from climate forcing, from this climate change that we're undergoing at this present time. Damage to crops is an example of corn, which is particularly vulnerable to heat stress, heat waves, and declining agricultural output productivity. How are these events linked? And if we can understand those linkages, can we make changes in different parts of that system to affect other parts of that system at different times and in different places? So, just by way of a little bit of background, plants are very much connected to climate, and indeed plants and climate are very much connected to each other, and this is one way to sort of visualize how profound that connection is. If we look at a landscape through a thermal image camera, you can see the canopies of those oak trees in this particular landscape that have their roots deeply embedded in the rock fissures of the landscape, attached to water. They're transpiring, they're cooling the canopy, and that canopy is several degrees cooler than the ambient temperature. The ambient temperature on that day was about 40 degrees, and you can see down in the riparian system down at the bottom where there's lots of water available, the canopy is much cooler than that, and the ground temperature, that bright orange, is much warmer than that. And so basically that principle, that applies across the global landscape. Different kinds of vegetation affect the climate in that way, with that mechanism by different amounts. So savanna systems or boreal forests, temperate forests, rainforests, their interaction with the climate and the climate affecting the behavior of those canopies and that heat balance and the evaporation from those canopies, they both affect each other, and they both change in different ways spatially and temporally across the globe. So as has been mentioned previously, one of the advances that has been made in understanding the Earth system as a whole is including in part of that system a better understanding of the way the vegetated landscape interacts with climate. And that's basically what I'm going to be focusing on today. The real crux of that problem, or that question of how that process works, really happens down at the cellular level on the surface of leaves through these very important structures called stomata or stomatal pores. And through that process, millions of tons of water is evaporated into the atmosphere annually, and with that the energy that's used to drive that process partitions solar energy into different aspects that affect climate differently. And so to understand that process at the global scale, we really need to understand also what's going on down at that cellular scale, because all of those cellular components, those stomata scaled up 1 billion times, is what's feeding back onto global climate. And just to emphasize just how profound and important that process is, these structures, these stomata, they evolved about 400 million years ago. We had a bit of a time series history of the planet Earth yesterday, but just to look at the period of history, the last 800 million years in which plant life has occupied the continents, the first 400 million years ago was pretty uneventful, and it was mostly plants that didn't have stomates, and basically the surface of the Earth went through these wet and dry periods because those plants were very poor at managing their water resources. But as soon as stomates appeared 400 million years ago, and they gained this control over their water resource, and they became very efficient at maximizing carbon gain for the amount of water that they used. And they were so good at that that kind of drove the evolution and appearance of global forests post-Devonian and into the Carboniferous. And in fact, it was mentioned yesterday that they were a little too good at this because they sucked out so much carbon from the atmosphere that they cooled the climate, and that had some downsides to it as well. So the question we're asking, back to this problem of heat and global warming and how that's going to affect processes and food security in the future. What happens when plants, when these canopies, whether they be natural systems or agricultural systems, when the canopy of those systems begins to fail and become stressed, because they're biological systems, so like all biological systems, they're vulnerable to breakdown under stress. And so we have in this basic concept of plants interacting with climate and these feedbacks going on. Plants affect climate, climate affects plants, and this has gone on for quite some time while we had a stable CO2, but now with that CO2 forcing, that's creating these two disturbances to that system. The CO2 is forcing climate, of course, but there's also a direct effect of CO2 on plants. Plants use CO2 for photosynthesis, and the plants are responding directly to that increased CO2 as well. So we have these two prongs of disturbance that are upsetting this feedback process that we have between plants and climate. So we need to understand more about how that process works so we can be better prepared to make predictions about heat waves and heat damage to crops, or perhaps even modify crops in the future to cope better with that. So the research we've been doing lately, and from funding from HFSP, sorry, Human Frontier Science Program, is how, well, it begins with, first of all, how stomates respond to CO2 and temperature. And so we know basically from observation, fundamental research begins with observation, and we know that if the CO2 is elevated, stomates tend to close to be more efficient at using water. They don't have to be open as far to get as much carbon if the CO2 is higher. And at elevated temperature, when temperature increases, they tend to open because it's advantageous to cool themselves. And so we wanted to know more about that process. And the first question we asked is, in fact, the first question I asked my molecular colleague in this process, I said, well, what genes do we have for understanding heat response of stomates? And he said, well, we don't really have any signaling pathways. We know about blue light, we know about CO2, but we really don't know anything about the heat sensing and response of stomates. And I found that rather remarkable. And that's an example of a classic example of a knowledge gap that needs fundamental research. And so we've begun to embark on efforts to uncover that pathway of heat response, and using some techniques called loss of function in plant models in Arabidopsis, we've begun to make headway into uncovering parts of that signaling pathway. And what we've found, I don't have time to go into the detail, but in principle what we've found, we've uncovered a new kinase that is part of that pathway, but what we've discovered is that receptor kinase actually is common to both the signaling pathway for CO2 sensing and temperature sensing. And so there's a little bit of a nexus there, and the problem is it creates this dilemma when both of those environmental signals are going in the same direction. When both are increasing, the plant is faced with this dilemma of whether it closes down to be more water use efficient or whether it opens its stomates to be a little cooler under excessive temperature. And that's probably not too much of a dilemma while temperatures are more moderate, but as soon as temperatures become more extreme, that then puts the plant into a different, a more challenging problem to solve, and more of a dilemma. And so stomates generally, much of our understanding of stomatal function is based on this principle of optimization of carbon gain. So there are lots of models that explain how stomates optimize that trade-off between gaining carbon and losing water. But we haven't really solved the problem of when you put heat over the top of that problem, excessive heat, how the plants cope with that. So that's an ongoing part of our research. The second part of our question is, how can we manipulate, if we know something about that control system, can we manipulate that system in a meaningful way that will give us information about global climate change? Can we implement that knowledge somehow in global climate models? And So global climate models, as you know, they run on these massive supercomputers, and buried within the thousands of lines of code, of course, are models. And all of the talks that you've heard today are basically models. All our understanding of the world around us is basically models, but we need good models and reliable models. And one of the questions that we're asking is, this model, so GW there on the left is basically the stomatal conductance. It's basically a representation of how wide open or closed those stomatal pores are. And this is a simple model. And the other thing is models need to be practical. So there's a trade-off between a really complex and accurate model and a model that's going to be malleable and amenable to usage in something as large and complex as a global model. So we are constantly searching for the optimum useful model. But just assuming this model, this model has its own, it had, like all models, it has simplifications, assumptions, but let's assume that it's an okay model for now. Most of the parameters in that model, all of them except one, are either climatic variables or biological variables. But there's one in particular that, and this is a common feature of many models, is that there's a parameterization factor. And the way that we currently parameterize that model to account for the differences, those differences in different type of vegetation around the globe is we change the value of that G1 constant. And so traditionally, that constant has been prescribed and based on very limited physiological data. So there are ways to go out with instruments and actually measure on a single leaf what the G1 value is for a particular type of vegetation. But the problem with that prescription approach is that that's very static. And when you've got a changing climate and a changing vegetation, with a static parameterization, how is that going to cope with the change, with simulating a changing climate? And so that's the other idea I wanted to introduce here is the models need to be realistic representations of a system. It's not enough for them just to work and give them the answer that you want. Now back to that parameterization feature of that model, what we've been able to do, well, it's been actually known that it's a somewhat enigmatic or mysterious parameter, but it's basically linked to the water use efficiency of plants. And if you were to map, so the water use efficiency is, in simple terms, is the amount of carbon that's fixed for a given amount of water. Or if you want to use rainfall as a proxy for how much water is available, and you plot productivity, global productivity versus rainfall, and what we see is across the globe, there's this non-linear response, non-linear relationship between productivity and rainfall. And basically that means that plants have different water use efficiencies in different world biomes. And so what you might expect if you were to visualize that, of course, in tropical rainforests, forests where water is abundant, the water use efficiency of those plants is quite low, and then plants get more and more efficient at using water the drier the environment gets, down to the most arid there in the left-hand corner. And so what we've been able to do is we've been able to develop theory and show empirically that there is a pretty good solid relationship between this G1 parameter and rainfall. And so when we use rainfall to parameterize, instead of doing it statically and somewhat subjectively, as we have in the past, if we take gridded rainfall and translate that to this sort of a continuous G1 value across the landscape and run it in global simulations, it actually, it not only works really well, so we've validated this with data, and it actually, the simulations work just as well and probably better with the use of this technique. So the example on the far right, so the left-hand example, try to go back. The left-hand example there is the default, and then we've got two different versions of increasing complexity of applying this mean annual precipitation technique. And you can see by this overall, this standard Euclidean distance is a metric that you can use to basically give you an overview of all the uncertainty and error in the model, in the simulation models. And you can see that the lower that value, the better the simulation. So we now have something more realistic. We have a parameterization process that is linked directly now to rainfall, so that when climate changes, that parameterization can move with that change in climate. And so that it's more in tune with our understanding of the way that climate and vegetation not only interact, but how they co-evolve together, whether it be over periods of millions of years or the next several decades. But the real value in this is we want to be better at projecting. We want to be better at understanding what the climate's going to be like, say, for example, at the end of this century or the next century, sometime into the future. And so we need reliable models, and we've done some simulations, some projections with this technique, and just by way of example, ask the question, well, how does that affect, for example, projections of heat waves? Heat waves are a very real threat in future climates. It was said 20 years ago by David King that record-breaking heat waves are going to be the norm. But we need to know when and where those heat waves are going to occur globally. So we need to get better at spatially and temporally predicting those. It's okay. I think we're all resigned to the fact that the climate's going to be hotter, but where exactly is it going to be hotter? And so with this new technique, I think we have to revise some of those projections. And for example, when we run, this slide shows when using that new rainfall-based method, it shows that there are some regions where there's improvement, where there's less heat wave days. That's the areas in blue, and there's some areas where heat waves are going to be more intense or longer. In this case, they're going to be several weeks longer, several weeks more days of the year where there will be heat waves. And we're talking about, with heat waves, these are lethal events. We're talking about the difference between saving lives or losing lives. So this, even changing this information by a matter of weeks will help in that pursuit, but also help direct resources, whether it be civil infrastructure, medical facilities, agricultural investments in agricultural techniques, biotechnology to genetically alter crops. Where should those efforts be focused more tightly? So basically, the conclusions from this, I'm just going to make some very overall general conclusions, and the first couple, of course, are obvious. Plant heat stress is a critical threat to food security, and plant regulation of water, carbon, and energy fluxes. We need to be better at predicting those processes. And large mechanistic gaps in this understanding can be filled using fundamental science. And so I gave two examples. So that the genetic pathway for controlling that stomatal regulation, that can be used as that is an example of the fundamental science. But also our models need to be conceptually realistic. So we need to sort of move away from sort of mistakes and habits that old habits that we're comfortable with, perhaps with older models, and think about new ways of using perhaps even those same models or developing new ones. And so basically this only will come through scalable and integrated approaches with transdisciplinary research and being encouraged by organizations like this to reach out and work across different fields with different people, perhaps ideally people that haven't worked together before. And so a big thanks to the Human Frontier Science Program and to my colleagues Sydney, San Diego, and Exeter, Stanford, Purdue, and NCAR, who are involved in just this specific project. Thank you very much.